Okay, so today we're going to continue our discussion about the biocatalysis in the part of the cofactor. So if you look at the cofactor, we actually have a definition of cofactor. So what is actually the cofactor? So cofactor basically is referred to all the non-protein molecule. Okay, bear in mind enzyme is protein molecule. So what is the cofactor? Cofactor is a non-protein molecule. They can be the inorganic compound, they can also be organic compound. Which is required to presence together with enzyme so that the enzyme can carry out their proper function. So that is a general definition for cofactor. So how many cofactor that we have? We actually have three type of cofactors that we learn, which is the meta-ion activator. So you can see here, you have the meta-ion activator, we have the coenzyme, as well as we have the prosthetic group. Okay, so yeah, there's three type of it. So meta-ion activator definitely is inorganic, and then another two coenzyme prosthetic group. Most of the time it is an organic compound, but there's also some cases there is inorganic compound in prosthetic group. Okay, so when the enzyme is actually when the enzyme is actually linked together with the coenzyme, they are actually called as the halo enzyme. Okay, part of the coenzyme function, they are actually supporting the reaction or the reactions that catalyzed by the enzyme. They can be either way become the electron carrier. They can be involved as part of the catalytic reaction, but sometimes they also involve as part of the active site. As you see in this particular diagram, they are actually part of the active site. And then sometimes they can also change the conformation, trigger the changings of the conformation so that the active site can fit exactly with the substrate. That one, the coenzyme will act as the activator. So different cofactors will have different function. So cofactors, they can be bind to the enzyme forming the halo enzyme. Okay, their role can be completing the conformation of active site. Second, they can also become the activator, which is activating the enzyme. They can also, being part of the catalytic reaction, act as the carrier of the electron inside the reaction or trigger the redox reaction. So this is some of the introduction. Okay, finish about it, we come to the activator. So we have a lot different type of activator. So in this particular case, we only learn about meta-ion activator. Bear in mind, when you go to university, there is a lot of meta-ion being part of the coenzyme. However, okay, I have to emphasize here, however, meta-ion activator, in this category of discussion, we only discuss those ion which is act as activator, which is activating the enzyme, changing the conformation of the active site here, changing the conformation of the active site so that they are ready to receive the substrate. Example, we have the magnesium, which is the activator for the hexokinase in order to run the process phosphorylation to glucose. Adding the phosphate group to the glucose become glucose 6-phosphate. There is actually a small mistake on the writing for glucose 6-phosphate. Bear in mind, it's supposed to be glucose, and then 6-phosphate. Okay, glucose 6-phosphate. Doesn't have any dash between the glucose and the 6. Okay, so yeah, this is one of it. And others example, do we have others examples on the meta-ion activator? So when we can actually see it, if you still remember, during your SPM, you're learning about the blood coating. Okay, blood coating. So during a blood coating, there is actually a lot of activator which is come out from meta-ion. So that one you can actually go back and find. So one of the tasks today, you can go back and find what is actually the meta-ion activator involved inside the blood coating. That we have a lot of different enzyme involved in that particular reaction. Coenzyme, the second type of the cofactors, they are organic compound. Okay, they are organic compound, bear in mind. All the all the coenzyme is organic molecule. So if I want to separating between what is meta-ions activator, what is coenzyme, the most significant difference is meta-ion must be inorganic compound. Coenzyme must be organic compound. Okay, so they are organic compound that required by the enzyme, because most of the time the coenzyme able to act as the electron transfer agent inside the enzyme. Okay, coenzyme, they can have several different functions. One of them, electron transfer agent. Another one of them, if you still remember during the previous slide here, you can actually see that the coenzyme here is actually to completing the active site. Okay, so how we actually know that they are coenzyme or not? So we look at two categories. One of them, what type of molecule they are, organic molecule. Second, we look at the binding. Does their binding is temporary? Does they bind loosely to the enzyme active site? If they are bind permanently, that is not coenzyme, that is prosthetic group. If they bind loosely, then that will be coenzyme. Okay, most of the coenzyme, they are vitamin, and they normally we're going to bring the word coenzyme with them. Examples like coenzyme A, and another coenzyme that you're going to learn in chapter 5 later on, the coenzyme Q, and the most important one is the NAD plus. Okay, bear in mind it's NAD plus, not NADH. Okay, NAD plus. NADH, I read this particular answer first because I not yet actually see the books actually write down NADH, so I just delete this answer first. But the NAD plus, yes, it is actually one of the example. FAD is not. FAD is permanently tied to the enzyme. So FAD is actually a type of prosthetic group. But the example of prosthetic group that we normally use is heme group lah. Okay, heme group. So what is the difference between coenzyme and prosthetic group? Prosthetic group can also be organic compound. They can be inorganic compound, they can be organic compound. Both is non-protein molecule. So the difference between coenzyme and prosthetic group is come from the permanently tied. Okay, prosthetic group is tightly bound with enzyme. They are not going to detach from enzyme. They are permanently attached with the enzyme, and they are part of the enzyme. Okay, they are part of the enzyme. So that is prosthetic group. So when we want to define, does it a coenzyme, does it a meta-ion activator, we look at them as bind loosely and temporary with enzyme. If let's say they mention, okay, they are permanently attached together with enzyme, then that one definitely 100% prosthetic group. Okay, example of prosthetic group is the heme group found inside the enzymes at the chapter 5. We have the cytochrome C oxidase, cytochrome C reductase. The cytochrome, inside the cytochrome we have the heme group. So the heme group is actually one type of prosthetic group. We also have the FMN, which is actually the flavin mononucleotide. Okay, flavin mononucleotide, that one is also one type of prosthetic group. And then we have the iron sulfur complex. The iron sulfur complex is also one type of prosthetic group. So we're going to go into detail on the examples of prosthetic group when we come to chapter 5 later on. So what I want you all to do regarding to this particular part is a revision note. Okay, a revision note, which is actually consists of the type of, okay, you can actually add in the type of cofactors here. So you have the coenzyme, okay, and then we have the prosthetic group, and then we have the meta-ion activator. Okay, then what you actually need to mention here is you compare in terms of what kind of compound they are. Does it come from the inorganic compound or organic compound? Then how does the binding with enzyme? Okay, so the binding to the enzyme, what kind of binding to the enzyme? And then after that, another one of it will be the examples. Okay, so this is the three main aspect that you must know. Okay, then function is not actually added here because I want you all to actually write out differently in order to separate the function with the example. Okay, so function based on different example. So example, you have coenzyme Q. If you have here coenzyme Q as one of your answer, then the function for coenzyme Q, what is it? And then if you have the example coenzyme A, then what is the function of coenzyme A? And then if you have the NAD plus, then what is the function of FAD plus? Same go for meta ion. If you write down the magnesium, okay, then you can also write down the for which particular enzyme, and then for this one like the copper feather copper, so what is the enzyme involved? So copper is actually very important in blood coating, so which enzyme inside the blood coating require copper, so write down. Then you have the prosthetic group example like the one that mentioned the heme group, the heme group in cytochrome C reductase. So heme group in cytochrome C reductase, what is their function? FMN, the flavin mononucleotide in the NADH dehydrogenase, what is their function? So write down in general function. So all this is separate from the table. So this is your revision note. Hopefully everyone can actually do it lah. If you're capable to do it, then is considered to be done. Okay, this is the explanation for FAD. FAD suppose is a type of coenzyme because they are organic compound. However, they are classified in prosthetic group by the scientists when the scientist is actually doing the classification because FAD do not move. FAD is permanently attached to the succinate dehydrogenase. So when they're permanently attached to succinate dehydrogenase, meaning